situation here. So today we'll continue with, of course, virology, the most exciting subject ever, at least as far as I'm concerned. Start out with what I often do is try and do a little bit of a review on what we've talked about in previous classes. So last time we talked about what is a virus, and hopefully it was clear that the definitions are not ridiculously obvious. I think the big thing there is what's the difference between a virion and a virus. And so thinking about viruses much more in terms of their life cycle as opposed to just the particle itself. Why to study viruses? Yeah, sure, viruses make people sick, but I'm much more interested in viruses for studying their ubiquity, their diversity, and really in terms of understanding viruses as tools. A lot of people talk about interdisciplinary science. Well, I think virology, just by its definition, is an interdisciplinary science because you can learn things about geometry, physics, immunology, biochemistry, molecular biology, genetics, genomics, you name it, in terms of things that epidemiology is also a good subject. So studying viruses, you can study all kinds of different things. We talked a little bit about virus origins. We'll talk a little bit more about that today or on Friday. Where viruses came from is a still very open question, and I've talked to a number of people, and myself included. If there's one thing I could do, it would be invent a time machine and go back to when viruses started and try and figure out what that is. So really interesting question. And of course, why viruses are important, because they're the coolest thing ever. What are we going to talk about today? We'll talk about really detection and counting of viruses. We talked about the ridiculous number of viruses we have here on this figure. All these little dots represent virions, or at least virion-like particles. Today we'll talk about the plaque assay. I actually brought a few plaques. Plaque assays, things that I've done in my lab. If I can do them, anybody can do them. And then look at how that actually works. The plaque assay is a really critical way for getting some data on how many active, and that is to say infectious, virus particles you have. You can also use plaque assays for that other stage of the virus life cycle, which is when it's actually infected a cell. To understand viruses, one of the big problems is that most viruses are pretty darn small. And at least so far, we don't have any techniques for studying individual virus, be they virions or infected cells. It's very hard to study those individual cells. So usually what people do is what's called a one-step growth curve, is looking at a whole bunch of viruses and virions doing the same thing at the same time, or more or less the same thing at the same time. So we'll talk about these one-step growth curves. They're an incredibly powerful tool to understanding the virus replication process. And then we'll look a little bit at mathematics. Everyone clear on the statistics? Poisson distributions, yes? We're all happy with Poisson distributions? Good, we'll talk about them much more. And the concept of the MOI, or the multiplicity of infection. And I'll mention this multiple times today. All that the multiplicity of infection is number of particles per number of cells. And so really quite straightforward. People get really confused. I'm not quite sure why. If we get a chance, then we'll talk a little bit about the virus replication cycle. This is a very general kind of what all viruses do. And multiple different steps. Turns out different viruses do these different steps in different ways. But every single one of them, because they're obligate intracellular replicators, I don't like to call them parasites, but obligate intracellular replicators, they've got to get inside a cell in order to be able to replicate. So we'll talk about that process, and then if we get a chance, again, we'll talk about virion structures, and that's why I have my nice little virus model here. Now, it may look like an underinflated soccer ball, but it's really a virus model. Just big picture kinds of things. Again, how many? We'll talk about the one-step growth curve, and then replication strategies and virion structures. A couple of years ago, some of the students asked me to put together some key concepts for any given lecture. These are basically it. So it's really, this is more for your hopeful edification, etc., rather than me going over them in any kind of detail. But it's a really great review kind of thing when those nasty exams come along. And speaking of exams, let's see how many of the clickers actually work today. So you are a junior biology major, senior biology major, post-bac, junior, senior, non-biology major, none of the above. And now I've got 35. That's like way better than last time. So good. It looks like they're actually working. So, and this, by the way, is counting down. And once it's counted down to the end, everything will stop. So. And I think we had something like 60-some people registered for the class. So maybe some of you have clickers that are not working, etc. Anyone tried to go and register their clickers yet? Good. Okay. I need to download and update those things. I don't have actually an official class list yet, so I need to get that before I can upload them. So now what I should really be doing now is counting the number of people to see, relative to the number of clickers here. But anyone else feel they need to vote? They're done, they're happy? We can just go ahead and stop. Okay. Are you guys curious? So mostly, oops, sorry about that. Showed up the results on the wrong screen here. Most of you are senior biology majors. So at least for the people who voted or admitted it. 15 are juniors, 15% juniors, 75% seniors, few post-bacs. These are the ones that are going to ruin the curve, so we'll ignore them. And then none of the above. I know at least one of the none of the aboves here. So with that, there's no correct answer to this question. Again, I'm only going to start counting them as of next week. So, can move ahead. Oops, go back to here. Wanted to talk about some of the breakthroughs due to viruses. These are far too many to mention, but it's getting back to that idea of using viruses as tools to understand basically what's being infected. All cellular life is infected, so we can learn a lot about all cellular life. And basically, almost everything we know about molecular biology comes, at least in the early days, from the studies of viruses. DNA is the genetic material, recombination, promoters, et cetera, et cetera, et cetera. Most of that came from the so-called phage group, which were a group of researchers starting about the mid-1940s that really decided that viruses were good tools for understanding the real fundamentals of life, and particularly people like Max Delbruck, in fact Salvador Luria, who I quoted a couple of times last time as a member of that particular group. More recently, people have been studying tumor viruses, and those tumor viruses are really wonderful tools for understanding tumors. So it turns out that they're important for some cancers, although probably relatively few of them. But it's in the study of these tumor viruses that a lot of eukaryotic specific molecular biology was discovered. Things like replication origins. Certainly in terms of cancer, oncogenes were originally found in these viruses, but then very soon thereafter shown to be present also in cells. But it was through the study of these viruses where they found those oncogenes in the beginning. And then, of course, retroviruses in terms of first things like HIV, etc. Turns out that a lot of the tumor viruses were retroviruses. So that's why people had already been studying these viruses before HIV came along, or at least we recognized that HIV was present. There are also some really cool applications of viruses. One of my colleagues up at Evergreen in Washington has been really one of the pioneers in something called phage therapy. You guys heard about? Oh, sorry. Yes, the phage group refers to bacteriophages, and I'll talk a little bit more about that as we move along here. Yeah, so basically when people talk about phage, it's always an abbreviation for bacteriophage. I'm in the process actually of writing a book chapter right now why you shouldn't call archaeoviruses phage. So, and generally eukaryotic viruses, people will almost never talk about them as phage. We'll talk about why they're called phage towards the end of the lecture today. So yeah, phage therapy, bacteriophage therapy. So the whole idea of bacteriophage therapy, people heard about it? Totally cool idea. Basically using the bacterial viruses to kill the bacterial pathogens. And so using basically something that's co-evolved for probably millions of years, again, we could argue about that, to kill off these pathogenic bacteria. And so there's a neat company called Gangagene, based on the water of the Ganges. The water of the Ganges has always been known as sort of holy water. You get better when you drink it. I would never want to drink the water of the Ganges, but it turns out it's loaded with viruses that infect the bacterium which causes cholera. So Gangagene is literally going and looking in the water of the Ganges to find bacteriophage that could work against cholera. Bacteriophage were originally found as and used as this kind of therapy, particularly for cholera, dysentery, etc. So it's kind of an old idea, but particularly with multiple antibiotic-resistant bacteria, particularly bacterial pathogens, it's really coming back. And so there's really great stuff up at Betty Cutter's website. Viral vectors, I would say, last time I checked, is about 80 to 90% of gene therapy uses viral vectors. As I mentioned before, one of my favorite descriptions, I should say, rather than a definition of a virus, is a bag of nucleic acid. And it moves that nucleic acid around. Well, if you're interested in gene therapy, genes are nucleic acid, and viruses are really good at moving nucleic acids around. So gene therapy is also a way. Nanotechnology. Turns out that the way viruses assemble into particular structures, not actually that dissimilar to something like a soccer ball, has really interesting implications, slow down, Stedman, implications for self-assembly nanotechnology kinds of things. Viruses have been doing this, again, probably for billions of years. This whole idea of putting together nano-sized particles, as we'll see, from very basic building blocks. And so if we can understand some of the principles on how some of these things are assembling, might turn out to be a really useful kind of completely technological, having almost nothing to do with biology. There's a nice article which I have posted on D2L called The Return of the Phage, which discusses some of this. So these are all sort of really big picture kinds of things. I could do whole lectures on each of these different subjects, but just wanted to throw them out there as kinds of things to think about. Now, why viruses maybe aren't all bad after all. So any questions about this stuff before I move into some more of the nitty-gritty aspects of things? Okay, so how many viruses do you have? This is the image that I had from last time. How many viruses were there in my little tube? 10 to the 11th of them. Well, how do I know that there are 10 to the 11th, in this case, infectious particles in that particular tube? Basically, through the Plaque assay, but that's only one way to know how many viruses, virus-like particles, virions you happen to have. Probably the gold standard in terms of how many infectious viruses you have is working with animals, certainly if you're talking about animal viruses. Animals have all kinds of problems. They're big, they're expensive, and it's really hard to get a decent number of them to get much in the way of an experiment. When you think about animal experiments, you're usually talking about 10 animals or something like that. So it's great if you're really interested in pathogenesis and how a viral infection really has something to do with an organism. It's almost impossible not to use animals. But if at all possible, try and move away from that. Electron microscopy, I showed you some pictures last time of some of the really amazing structures that you have on some of these virions. And also electron microscopy is a great tool for looking at the infection process. So you can see the cells and the virions at the same time. But electron microscopy has two actually really big negative aspects to it. One is you're just seeing particles. You don't actually know if they're infectious. And the second one is they're really low throughput. You can't look at, you're not going to count 10 to the 11 particles in an electron microscope. And anyone who's done electron microscopy, I know a few of you have, knows that it's really not quantitative at all, at best semi-quantitative. So what most people do in terms of working with viruses is a so-called plaque assay, which is shown up here in a diagram, and those of you who are in the recombinant DNA lab will actually be doing these later this afternoon, where you take a virus suspension up here at the top. This is using a particular amount, doesn't really matter. What you have, and this would be either infected cells or individual virions. You do serial dilutions, and so this case is just 0.5 mils to 4.5, so it's a 1 to 10 dilution. Multiple serial dilutions. You'll notice the numbers here get up to pretty high dilutions. So in this case, it's a 10 to the 8th. And then what you do is you take that mixture now of, the diluted viruses, mix them with susceptible cells. And the real key here is it's a vast excess of those susceptible cells. You really want to have mostly non-infected cells, but a few of them are actually going to get infected. Then you incubate these two together, usually on a Petri plate, and what happens is you'll get, for each of the infectious viruses, or virions for that matter, could be an infected cell, you should get one spot that happens on this plate. Well, why is that? Well, because what's happened is that particular individual infection, that individual infectious particle, now has infected the cells right next to it, which have infected the cells right next to it, which have infected the cells right next to it. So you have to do this for quite a long time, multiple replication cycles. And then you just literally count the number of spots that you have on a plate. And a lot of the eukaryotic viruses have very interesting modifications of this, but it's basically exactly the same thing. You're looking for multiple rounds of infection in one particular place, which you can then back calculate, because you've done this 10 to the 8th dilution. You've got an average of 12 spots on your plates. 12 times 10 to the 8th is 1.2 times 10 to the 9 in terms of what you started with way back there in terms of your virus suspension. Another way of looking at that is here. Again, mixing together your phage dilution with, in this case, bacterial cells. Put them together with top agar. The critical thing with top agar is it basically slows down the cells. They can't run away. Because if they're motile and moving around, then you're not going to get the virus actually confined in one particular place. Pour that mixture onto a plate, put your top agar together, incubate, and then you look for plaques. And these are those plaques right here. Here's a nice example here of a plaque on a plate. And the reason, getting back to your question about phage and bacteriophage, this was thought to be the viruses eating the bacteria on the plates. And phage comes from phagos, the Greek to eat. So these were things that were eating bacteria. So the bacteriophage. So each of these represents a single viral infection event. Any one of these plaques actually now represents probably millions of virus particles because it's gone through all these multiple rounds of replication. You wouldn't see a single infection. Here's an example of some plaques which I did in the lab. This is from a virus called Plaques-174 that, again, the people in the recombinant lab are going to be working with this afternoon. This is pretty typical in terms of the number of plaques that you get. 67 plaques up there, 81 down here. You'll notice that these guys often, kind of run together, and actually doing accurate counting is problematic, particularly when you've got larger numbers like this and larger plaques that form. So it's really important that you do multiple plates, and so you can actually average across these because you'll have lots of different counting errors, etc. in that process. So, and again, I brought a number of plaques here. There's one that you can come take a look at later. This is one of my favorite numbers to count. I always call it TMTC. You know what TMTC stands for? Too many to count, exactly. And then some of these here. These are some from bacteriophage T4. 297 on this one. This one, I think, was a TMTC. This one's way, way, way too many to count, etc. But depends. And I also wanted to bring these two. These, the T bacteriophage, and I can just back up here real quick. These usually give these really tiny spots, whereas other bacteriophage will give you much larger plaques like Plaques 174 on this case. And so, about 100 is about the maximum you can count on this plate, whereas you can easily get a couple hundred on those. Yeah?
If you get too much to count, do you go back to further serial dilutions and then do it again?
Yes, or you do like I did here and do multiple different serial dilutions. So this was a 10 to the minus 4 that I plated, 10 to the minus 3, 10 to the minus 5, 10 to the minus 6, and then you basically bracket the range that you think you're going to find, and then you'll just count the ones that are countable. So means that you often end up with a whole bunch of plates. This particular assay that I did, I think I had something like 36 plates that I was looking at. So yes, statistics good for this, this process. So that's kind of why we're also doing it, hopefully multiple plates in the recombinant DNA lab. All of you, how many people actually are taking that lab right now? A couple of you? Yeah, so hopefully you'll get lots of pretty plaques like this. Plaque assay is by no means the only way to study the number of viruses. I'll talk about a couple of different ones now in the next couple of slides. Some viruses don't plaque very well, and that usually has to do with the hosts that you're working with. So bacteria are great because they make really nice lawns, they grow nicely on plates, etc. Animal cells, on the other hand, are usually a lot more finicky. And so sometimes it's really tough to find them. You also have to find cells that that particular virus is going to infect. And so that's another issue that sometimes is a pretty big problem. Another way to deal with this, many viruses, and we'll talk about these a lot more later on, have what's called hemagglutination molecules on the outside of the virus. And that's been taken advantage of with the so-called hemagglutination assay. This takes advantage again of those molecules that are on the outside of the virus, which bind to, heme, and actually in this case it's just literally red blood cells that they're binding to. They bind to sialic acid, which is on the outside of many cells, particularly red blood cells. And basically all that happens is you get your red blood cells that you put together with virus. If you have virus in there, what happens is the virus binds to a red blood cell on one side, but the virus has multiple of these hemagglutinins, and it basically just cross-links between two red blood cells. And so in the case of virus, you have this clumping that takes place. If you don't have virus, then there's no cross-linking that takes place. These guys are not in a sort of a cross-linked network. They end up being kind of a spot down at the bottom. And so basically all you need to do here is, again, dilutions. It's an example of one of these dilution assays right here, going out to 1 to 8,192. You'll see here there's nothing. And that means we've diluted out beyond the hemagglutination assay sensitivity here. And as you go down in terms of your concentration, basically here about 10, 24, 512, etc. The very low dilutions, you get nothing. This is a negative control down here in line G, and a positive control here in line H. So there's one thing which may be a little strange here, and that is when you do these very low dilutions, in some cases you'll have, look, hemagglutination assay activity, but in very low dilutions it looks like you don't have any hemagglutination activity. Any idea why that might be? We'll talk much more about this later on. It turns out that some viruses, influenza in particular, which is mostly what this assay is used for, as well as having a hemagglutinin molecule on the outside of the virus, also has a neuraminidase, which you may have heard about. Well, what neuraminidase does is it chops off the sialic acid on the outside of the cell. So if you have enough virus which is there, it's done this clumping and cross-linking, but then it chops them apart. And so that's why here at the low dilutions, high virus concentration, you see that there's those. Again, so you've got to do dilution series of these to get an idea what's going on. This is very often done for clinical samples. You're basically looking yes, no, is there some kind of hemagglutination activity which is present in a particular sample. There's some advantages to this. It's really fast, it's really cheap. You can modify it, you can use antibodies then to various different cell types, but the major problem is the sensitivity is actually really, really low. If you think about 1 in 8,000 relative to the 1 in 10 to the 8th, 10 to the 9th that we're looking at with plaque assays, this is clearly an issue. So this gets back to one of the really important points about this whole virus counting business, and even true for plaque assays, and that is that the number you count is often way lower than the actual number of virions that you have in that solution that you started with. So there are a couple of reasons for that. One may be that you just don't have a high enough sensitivity in your assay. There could be some technical issues. But often you find that you're just counting particles, and again, this gets back to why electron microscopy is not such a good way to count viruses, is that some of these virus particles might not be infectious. So, in the case of HIV, for instance, if you look at just count number of virus particles versus the number of infectious particles, it's about 1 in 10,000. So, very, very low infectivity. You could have a particle that looks just fine, but its genome has problems. And because its genome has problems, then it can't actually replicate. That nucleic acid that's been packed inside the bag is non-functional. You can also have lots of empty capsids. This, it turns out, is the case for HIV. And then depending on what cells you're using, or maybe if it's an animal assay, for instance, antiviral defenses, which may be taking over. And this is not just true in animals. Turns out that bacteria. Has anybody heard about CRISPRs? Yeah, so turns out there's an adaptive immune system in bacteria. We'll talk about that later on in the class. Really fascinating system. So may turn out that the cells you're trying to use have some kind of defense mechanism. So you're going to underestimate the numbers that you actually have. So numbers of viruses, important if you're worried about infection, but also it's really important to trying to do studies with viruses. And the reason for that has to do with this so-called one-step growth curve. Again, the big problem, although we're getting to the point that we may be able to actually study single virus particles and single virus infections, but still not straightforward. So this is the standard way that people do this. Basically, what you want to have now is a population of cells that are infected with viruses at as close to the same time as possible. So the idea here is to have a simultaneous infection, which is then hopefully synchronized. And so you can follow a population of infected cells. And you follow that population of infected cells, and then the virions that are being produced by that population of infected cells. And then you can back calculate and say, well, each individual virus is replicating in this way. It's definitely an oversimplification, but it's worked extremely well. So the idea is you do a simultaneous infection, you cycle this replication cycle for viruses. Actually, I should modify this. Turns out that some viruses can go through their whole replication cycle in about 5 minutes, or in some cases it takes days. Critical to be able to do this experiment properly, because you want to have simultaneous infection, you want to make sure that all of the cells in your assay are infected. And to do that, you need a high multiplicity of infections. So lots of infectious virus particles, again, relative to the number of cells that you're infecting. So you do this simultaneous infection. This is a classic example of one of these one-step growth curve assays. You infect with usually an MOI of about 10, and then you follow virus production, RNA production, DNA production, protein production, virion production over time. And so that's all that this one-step growth curve is about. So If you look at this particular example, this is from the textbook, do your infection, the number of infectious virus particles goes down because they're, well, infecting cells. We mentioned before, you're not reusing those individual virion particles. That then we're just following the black line here. That then, after a certain period of time, then will pick up, and eventually you end up with a large number of virus particles. You've gone from here, and again, this black line here is your PFUs. So you're just doing a plaque assay. Start out here, MOI of 10, and just over time you're doing your plaque assay, and eventually that picks up. So that's classic one-step growth curve. You're looking at plaque assays over time. You're looking at how virus production is happening. But virion production, or infectious units, is only one part of the process. There are lots of other things that are going on there. In this particular case, this is a tumor virus. If you look at the viral messenger RNA, it comes up here, starts to be produced after a certain period of time. Then much later, you start to get the viral DNA being produced. Then you start to get the viral capsid protein. So viral DNA, excuse me, is this solid brown line. Viral capsid protein is the dashed line. And then eventually you start to get virus particles. We'll look at a couple more examples of these. This is a slightly more simplified one-step growth curve. Again, virus to host cells, get rid of any of the uninfected cells, not uninfected, the only infected cells, and then just do the plaque assay over time. There's one slight difference here, and this particular assay, you also can do an artificial cellular lysis, and the lysis of cells, basically this will give you intracellular infectious virions. And so you can do these kinds of one-step growth curves just looking at what's in your supernatant, acellular, or viruses which have already been produced, or say infectious virions, which are present inside the cell. If you look at this process, again, this is a completely generalized one-step growth curve. You add viruses, you start out getting some of the early proteins. Usually these have enzymatic activity. Then you have production of nucleic acid. Then you have production of capsid. And then you put everything together and they get out. So this is the process. This is also known as the eclipse period and maturation period. The eclipse period right here is when you don't have any virus particles that are around. And so it's really important. This is where all of the interesting molecular virology is going on. It's in that attachment and absorption phase right here when you're doing the infection and making all of the virus particles. We're going to concentrate 95% of the rest of the class on this part, because this is how you're getting these viruses put together. Some real growth curves are shown here on the next slide, which also reminds me, these are figures from the Flint textbook, which should be on reserve in the library now. I took it over there yesterday, and usually takes them a day or two to actually get that. So it's on 2-hour reserve over there. So these are just some cases here. 2 different viruses at the top. This is just a bacterial growth down here at the bottom to show that they're different. Adenovirus type 5, that's a DNA virus. It's a non-enveloped virus. Western equine encephalitis virus here is an enveloped virus, has a lipid membrane around the outside. We talked about those before. If you look at the top one here, just looking at the either total viruses, which is the blue dots, or the intracellular, which are the red dots, those are basically identical to each other. Starting out with their PFU per mil now. This is PFU per mil instead of PFU per cell. So again, it's a plaque assay. Go down at the beginning. That's when you're having your infection and absorption process. Stays flat until you start to build up virus particles. In this particular case, again, this is a naked virus. You see there's a difference between the red virus particles and the orange line. And that's when you've done the 2 separate one-step growth curves, one where you've done lysis of the cells, the other one where you haven't done lysis of the cells. And what you can see is there's actually a time lag between the two. 12 hours that you're starting to see virus particles inside the cell, and 16 on the outside of the cell. And that process there, that 4-hour time difference, actually tells you a lot about the replication of this virus. You have to get a certain amount of virus before it bursts open the cells and can get out. And so just that process is telling you that you've got a virus which is forming inside the cell. Usually these are going to be non-enveloped viruses, because most enveloped viruses are picking up their lipid on the outside of the cell. And you can tell that by just looking at this assay here. For the enveloped virus, production of virus intracellular and extracellular is exactly the same time, because these are now being made. When you do your intracellular, that's just a lysis step of the cells that you do artificially. So artificial lysis gives you the same thing, same timing here for the enveloped viruses, particularly those that are getting enveloped at the plasma membrane. And then differences for the naked viruses, you have a time delay here. Yes?
What did PFU stand for?
PFU, plaque-forming units. So infectious virus particles. So I mentioned that before. Thanks for pointing that out. So again, how do you do these? MOI of 5 to 10. We'll talk about why you need an MOI of 5 to 10 in just a second. There are two things that a lot of virologists talk about: the eclipse period and the latent period. Eclipse period is when you see no virions anywhere inside or outside the cell. And that's here for your adenovirus type 5. It's a 12-hour period. The latent period is no viruses on the outside. And so if you see a difference between those two, the eclipse period and the latent period, that tells you that there's a certain amount of virus particles, virions, that need to be accumulated before you get lysis. If, however, the eclipse period and the latent period are the same, which is what you have on a lot of these enveloped viruses, that gives you at least an indication that it's probably picking up something from the cell, which is really important. And that lysis step is not a separate step which is going on. A lot of people also talk about the so-called burst, and this is just the number of PFUs, plaque-forming units, that you have at the end divided by the number of PFUs that you have in the latent phase. So, latent phase, you see it's not 0. Why is it not 0? I heard somebody muttering, but it wasn't loud enough for me to understand it. Yeah?
Because there's viruses inside the cell.
Yeah, so the reason for that is that you have infected cells there. And so this number, actually, the PFUs that you have during the latent period, those are all the viruses that are in their cellular part of the life cycle. And you remember in the plaque assays, you're going through multiple rounds of replication. And so there's plenty of time for whatever genome was in there to replicate enough virus particles to go through. So that's due to infected cells. And so just by doing these one-step growth curves, you know how many infected cells you have, you know how many you end up with, and that gives you how many virus particles are being produced per cell in that one process. So the burst is also a very important aspect of what's going on in terms of understanding different viruses. Different viruses have different burst sizes. Burst size can be as low as 5 to 10. It can be as high as a couple thousand. In some cases, maybe even more than that, which is kind of amazing, but yeah, it's a different story entirely. So what about MOIs? And why do I keep beating on this whole idea about MOIs? Well, to do one-step growth curves, you have to have a high MOI. Why do you have to have a high MOI? Well, that's because the interaction between viruses and cells, remember, these are populations. They're not individual particles that we're talking about here. They're random interactions. And so that random interaction is really nicely described by a Poisson distribution, which is down here at the bottom. Don't worry about memorizing the formula. If I give you something like this in an exam, I will give you the formula. Don't worry about it. All it is the fraction of cells that are infected by or just interacting with k different particles is going to be e to the minus m, and m here is your multiplicity of infection, so number of virus particles divided by number of cells times m to the k, and then k is that number of particles, divided by k factorial. Now, what does this really mean? I put together a little diagram of this. If you have an MOI of 1, and this is 5 virus particles and 5 cells, they do this infection. What you'll see is that some cells are going to have 1 virus infecting, some are going to have 2, and some are going to have 0. And this is, again, just random distribution. If you do an MOI of 2, you're going to have some cells that have 1, some cells that have 3, some have 2, some have 1, and some are also going to have none. And an MOI of 5, so we've got 25 virus particles, 5 cells. You'll have some that have more than 5, some have less than 5, but you're getting closer and closer to have all of them being infected. And so that's basically all that this Poisson distribution means. There's actually a quite nice discussion of this. If you go to Google Books and search for Poisson distribution virology, you can take a look at it. But this is just plots of that function for any given, in this case it's A, but basically it's the same K. So if you had a M of 2, we're looking now at number of, or percentage I should say, of cells that have a particular number that are infected with them. So we'll just look at this black line here. You'll have a certain percentage here. So this is about 0.5%. It's actually about 0.36%. We'll look at those numbers a bit later. So here, at MOI of 2, so twice as many virus particles as you have cells, you still have quite a few that are non-infected. Here, you've got quite a few that have 1, but that's not 2 which you had. Quite a few that have 2, but again, this is still way less than 50%. It's about 26%. And then less and less that have more and more. The higher MOIs that you go to, higher M's, you end up with, again, this distribution around where you started, but very small numbers out here at your non-infected. So you can actually look at the numbers here. P of 0, which is usually what we care about, that's the non-infected proportion of cells. If you just mix the same number of virus particles with the same number of cells, 37% of them are going to be non-infected. Okay, if you're trying to do a synchronous infection, this is clearly a problem. MOI of 2, 13%. MOI of 5, still about 1% of your cells are going to be non-infected. So to do a nice synchronous infection, people try and do MOIs of 10. And anybody who's working in my lab or any other virology lab knows that getting MOIs of 10 are a real pain. Getting enough infectious virus particles is a real drag. But you want to get as close to this as you possibly can. So P of 0, again, the probability you're going to have uninfected, it goes way down relative to the numbers of your MOI. A lot of people also like to think about, instead of the amount uninfected, is the amount infected, and that's basically just 1 minus this P0. So proportion that you have. It's just from 0 to 1. So 1 minus that is going to be everything else. Again, so an MOI of 1, 63%, infected, 2, 86, etc. So again, MOI of 10, 99.995, which is there. So any questions about either these one-step growth curves or the MOI Poisson distribution? Again, I will give you numbers if you need them, and if you need a calculator on the exam, I will let you know. Don't expect you to do them all by hand. Okay, good. So let's move on and talk, again, generalizations about all viruses. All viruses have to go through these steps, and basically this is the cartoon up here. I've given you a little bit more detail here. We'll go through all of the different viruses in the course and talk about each and every one of these individual things, and you'll see the outlines at the beginning of most of my lectures. They'll have those individual pieces. Which reminds me, I haven't posted last year's lectures yet, but I'm going to try and get them. I talked about putting them on D2L. It's going to be a mess, so I'll put them on YouTube. So they'll be there. First thing that happens up the top, binding to a cellular receptor. So that virion, alive or unalive, we can argue about that later, has to somehow find the cell, because these are obligate intracellular replicators. So has to find the right cell, so you have that binding. Turns out that virions can bind to pretty much anything that's on the outside of a cell. Proteins, lipids, carbohydrates, you name it, on the outside of a cell. And we already saw in terms of the hemagglutination assay, the sialic acid is a carbohydrate, which lots of viruses bind to. What cells an individual virion binds to is called tropism. And so it's that number of cells that have the particular extracellular molecule that that virus binds to, that's going to give you your tropism. Once you've bound, that's only the first step. Again, these are obligate intracellular replicators. So you've got to get inside the cell. Lots of different ways. That's number 2 here. Also, you need to release your genome from that capsid that you have. All kinds of different ways that this happens. And again, we'll talk about them in some more detail. Some of my favorite is really kind of literally a drill that drills through the membrane and then releases the genome on the inside. Probably most of you have seen the T4 infection video ad nauseum, but I'll show it again because it's so totally cool. You also often will have some damage of the cellular membrane on the outside. Many viruses have enzymes that are part of the actual capsid on the outside, which will degrade part of the cell in order to be able to get in. If you are a enveloped virus, one of the great things about having a membrane around yourself, most cells have membranes around them. Membranes love to fuse with each other. So that's a really easy way to get inside the cell. And then once you get inside the cell, then you've got to release your genome. So in many cases, you have degradation of the capsid. And that can either be a cellular-mediated degradation or, in fact, a virus-mediated degradation that takes place. Once you have released your genome inside the cell, then you start to make virus-specific proteins. So usually this is split up into early, sometimes middle, sometimes late, but there's always early and late gene expression that you have in different viruses. So we'll talk about those in a lot more detail. Particularly the early genes are going to be involved in making more of the virus genome. We continue here. These, again, mostly involved in genome replication. We'll talk about S phase for those eukaryotic cells that are undergoing the cell cycle. Then you have late messenger RNAs. These late messenger RNAs, late proteins, are almost always made after you've undergone genome replication. So the early genes are going to be those that are made from that genome which gets released in the first place. And then the late genes are going to come up later on. If you remember way back to that one-step growth curve, the first one that I showed, you saw viral mRNA taking a little blip and then being stable and then going way up a little bit later on. It was that early blip which was the early virus genes. And then the late messenger RNAs were the continuation thereof. And we'll look at those in much more detail. What do most of these late messenger RNAs code for? They're usually things that you need to make virions with. So there'll be structural proteins, scaffolding proteins that we'll talk about later on. These are proteins you need to make virions but don't actually end up in the virion itself. And then often how you can get out of the cell. And then many of these late proteins are also involved in the very last step, which is getting those virions outside the cell. In some cases, that's busting open the cell completely, but that's not true for all viruses by any stretch of the imagination. Budding is a really common way that viruses are released, or virions, just say, are released from the cell. In some cases, virions are transported directly from cell to cell. They don't even actually get released from a cell because most cells in a multicellular organism are right next to each other. So it turns out you don't even have to get outside of a cell per se. You can just hop into the next cell. We'll talk about some of those later on. And then in many cases, there are other cells or organisms that move viruses around. And so these are viral vectors, like my two, I think, really cute daughters at the beginning, moving viruses around. So certainly other ways that these things happen. So that's the process. Again, the whole class in 2 slides. But we'll talk about some of the more details about that a little bit later on. One of the things I mentioned last time in terms of virus definitions is all viruses that we know of are dependent on cellular translation. And what that means is that the real critical thing about virus replication is going to be how does it make its messenger RNA? Because, again, everything is dependent on cellular translation. No ribosomes, so far, have been found in viruses. So you have to get a messenger RNA, and by definition, and we'll talk a lot more about this as we move on through the course, your messenger RNA is positive strand, because that's what's coding for your proteins. So positive strand RNA, your messenger RNA. It turns out that there are some viruses that their genome is positive strand RNA, so that can work as a messenger RNA in and of itself, which is great in terms of translation. Of course, in terms of replication, there's some issues with dealing with that. But getting to that messenger RNA is dead easy, because as soon as you release your genome, you've got a messenger RNA. That's only one of about 6, or maybe slightly more than that, of the different viruses that are out there. Classic, and in fact, this is what most of the phage group was working on, because they're interested in cellular molecular biology as well as virus molecular biology. Many viruses have double-stranded DNA in there, packaged inside their capsid. These then have DNA-dependent RNA polymerases, either virus-encoded or host-encoded, which can then make messenger RNA. So just like what happens in cells. So these guys are relatively easy to understand. And again, I mentioned before, many viruses have these positive single-stranded RNA genomes. But there also are a few other bizarre different ones. One that we'll talk about right towards the end of the class actually has a mixture of single-stranded and double-stranded DNA that's packaged. There are a lot of single-stranded DNA viruses, which I'm getting really excited about, because our lab has discovered some of these in the last couple of years. Turns out lots of those little dots that you see in those fluorescent dye stains of the virions, many of those dots are single-stranded DNA viruses. And in fact, as you mentioned, I think one of you asked the question last time about how many of those are there, and it turns out you can't detect a lot of the single-stranded DNA viruses, so there may be way more of these out there than we ever thought. Single-stranded DNA viruses are really interesting, because nothing in the cell can do anything with a single-stranded DNA. So it has to go through this double-stranded DNA intermediate in order to make messenger RNA. And it seems like a really convoluted process to go from single-stranded, have to go through double-stranded before you can make the messenger RNA, before you can make the proteins, to make more of the DNA, to make more of the virus particle. So as if that wasn't crazy enough, in the mid-1970s, David Baltimore and Howard Temin, at least theoretically independently, discovered the retroviruses, which actually in their bag have positive strand RNA. But what's really bizarre is it doesn't replicate by using that RNA directly, as it could, because this is actually coding RNA that you get in these retroviruses. It goes through a single-stranded and then a double-stranded DNA intermediate, and then finally makes its RNA. And we'll talk about those towards the end of the class. So these are the single-stranded DNA viruses, double-stranded DNA viruses, retroviruses. But just to add even more interest to this, it turns out that there are also a number of virions which package negative strand RNA. And it turns out these are a lot of the really nasty viruses, at least as far as humans are concerned. Ebola virus, influenza virus, lots of these are actually negative strand RNA viruses. And all that negative strand means is that's not actually coding the proteins. So you have to have a RNA-dependent RNA polymerase in order to make this messenger RNA that you need to make more viruses with. And that process, making that viral RNA-dependent RNA polymerase, means that any single-stranded negative strand RNA virus, does it have any way of coding for early proteins when it comes inside the cell? No. So it actually has to make the messenger RNA before it can make any viral proteins. And what that means is that all of these negative strand RNA viruses have to have an RNA-dependent RNA polymerase with them in the virion. Because they don't have it, they're never going to be able to replicate. Same thing is true down here for the retroviruses. They have to have a reverse transcriptase. Otherwise, it's not going to be able to be functional. So you always see the pictures and we're releasing the genome inside the cell. Well, it turns out in the case of these negative strand RNAs, and it turns out the double-stranded RNA viruses as well, all have to have viral-dependent polymerases, and the same thing is true of the retroviruses. So another way of looking at this is the so-called Baltimore classes. It's basically exactly the same thing. It's all about the messenger RNA. I'm not going to expect you to remember the numbers here, but basically there are 6 different kinds of genomes that can be packaged, either double-stranded DNA, single-stranded DNA, the retrovirus process, this positive strand RNA, which can be used directly as messenger RNA, but in order to make more of that genome, you actually have to use some negative strand as well. The negative stranded RNA we just talked about, and double-stranded RNA, which actually turns out to replicate a lot like these negative strand RNA genomes. So that's basically it for Chapter 1, at least from the textbook. We talked about what a virus is, obligate intracellular replicator with an extracellular form, or bag of nucleic acid, which of course is ignoring the whole cellular replication process. Why we should bother, hopefully convince you that there's some reasons we should be bothering about virology. Originally discovered, of course, because there's really small particles. They could go through filters but still cause disease. How many viruses? We mostly talked about the plaque assays, and again, I've got the plaques up here at the front. And then we talked about the various different Baltimore classes and virus replication cycles. I think we'll stop here. Instead of talking about virus structure, it'll give you a chance to come down and check out some of my plaques down here. Again, T4 over here. This one's actually a Phi X174. And so on Friday, we'll talk about virus structure.